Hello, everyone, and welcome to our webinar entitled Diversity and Inclusion in the Microbiology Classroom, hosted by Wiley and the American Society for Microbiology Press. My name is Megan Angelini. I'm a Managing Developmental Editor for ASM Books, and we're so pleased that you can join us today. This webinar will explore ways to foster diversity and inclusion in microbiology while stimulating learning and facilitating classroom discussions using elements from the new third edition of our Microbe textbook. Before I hand it over to our speakers, I do want to note that you're in listen-only mode to avoid any background noise. However, we invite you to type any questions you might have into the question box in your control panel so that the presenters can address them during the Q&A portion of the webinar. This session is being recorded, and in a few days' time, you'll receive a follow-up email with a link to the recording. Additionally, you'll find a copy of the slides in the handout section of the webinar panel. So please allow me to introduce our speakers for today. Dr. Michele Swanson is a professor in the Department of Microbiology and Immunology at the University of Michigan Medical School, where her research group investigates how metabolic cues govern Legionella pneumophila's resilience in the environment and virulence in phagocytes. And she recently served as president of the American Society for Microbiology. Dr. Elizabeth Joyce is a professor in the Department of Microbiology and Immunology at the University of California at San Francisco, where her primary role is teaching microbiology and immunology in the schools of medicine, dentistry, and pharmacy. And Dr. Rachel Horak is a senior specialist in the education department at the American Society for Microbiology, where she facilitates an international community of microbiology educators with the goal of increasing diversity, equity, and inclusion for all undergraduate students. Together, they are the authors of the new edition of the Microbe textbook. This third edition of Microbe builds and improves upon its best-selling predecessor with case studies, activities, and features to engage students and expose them to the breadth of diversity that exists throughout the field of microbiology. The authors have paid special attention to instructor support materials. Adopters of this new edition will have access to curriculum guidelines and learning outcomes, answers to end-of-chapter questions, and a large peer-reviewed test bank. Adopters' materials also include lessons plans and exercises and activities that instructors can use to enliven their classes. There are a wealth of supplemental resources, including exercises and other activities ranging from podcasts and videos to blog posts that enhance remote teaching and learning. So without further ado, please allow me to hand it over to our presenters.
Great. Thank you, Megan. Hi, I'm Rachel Horak. I'm going to speak to you today about a couple of new and unique features in Microbe 3rd edition. Today's presentation, we're going to talk to you about 4 main features that we included in Microbe 3rd edition. First will be elements for increasing diversity, equity, and inclusion. We also call it DEI through this presentation. And we're going to tell you a little bit about our brand new instructor's manual. We're going to talk to you about some of the new elements in scientific content and how we bring that content to the students. And then lastly, we're going to talk about student-facing elements for learning. So let's go into these elements for increasing diversity, equity, and inclusion. I'm willing to bet that all your institutions are requiring educators to address DEI in the classroom because an inclusive and equitable teaching for student populations is simply good teaching. Knowing that DEI is essential to good teaching, we intentionally designed elements and wrote in an inclusive fashion. And representation matters. Published studies indicate that stories can encourage a sense of belonging in students. Ask many students what a scientist looks like, and they're likely to say white, male, able-bodied, because for too long, these adjectives described who got to be a scientist, but not anymore. We see examples of this every day in our institutions. So in Microbe 3E, we found a wide diversity of scientists to tell their story to us. One example here you can see is Kyle Card. He is a disabled scientist with a rare disorder. It's a neurological condition called Mobius syndrome. He told his story to us. And we hope by reading about scientists who happen to be disabled or LGBTQIA+, or Black, or US immigrant, students will appreciate the human diversity in STEM and find a sense of belonging, and therefore continue in the field of microbiology. Next slide. Microbe also has features that address public attitudes and policies that have traditionally impeded not only scientific progress, but also equity. Here's one example on the right. You can see we included new Microbes in Society feature boxes throughout the textbook. This one example talks about how marginalized populations have been blamed for disease spread before the advent of germ theory. This one example is from the Jim Crow South in the early 1900s, and it's a very racist political cartoon that blamed Black citizens for spreading disease. And we think that it's important to remember these histories so that this doesn't happen again. Next slide. Microbiology impacts and its impacts know no geographic boundaries, and neither do undergraduate readers of our textbook. So we were very intentional to increase examples of microbiology from non-Western cultures, as can be seen here in this example of a fermented beverage from a Xhosa culture in South Africa. We were also very intentional with our written language to ensure that it was inclusive and culturally competent for students from all cultural backgrounds. For instance, we did not assume that students were familiar with Eurocentric fermented foods, such as sauerkraut. Next slide. Now we're going to talk to you a little bit about our brand new instructor's manual, which I think will be very useful to all of you because all of you are educators. And this should help you save class prep time and design better courses for your undergraduate students. So let's take a peek at some of these new and unique aspects of the instructor's manual and the resources that we give you. The entire book is centered around the peer-reviewed ASM curriculum guidelines for undergraduate microbiology. These guidelines were published in JIMBY, which is known as the Journal of Microbiology and Biology Education, in 2012. These curriculum guidelines were the result of input from dozens of educators and scientists and presents the 27 fundamental concepts and 6 core themes. Together, these curriculum guidelines are a fully comprehensive view of the entire field of microbiology. All concepts presented in Microbe 3E are mapped to these curriculum guidelines, and we indicate all the key concepts at the front of the chapter. You can see an example of this from Chapter 3, the microbial cell interior, is not just about structure and function, but also about the core concept of information flow and genetic core concepts. Next slide. After all of your efforts to teach your students, how do you really know that they are learning microbiology concepts? Do you have evidence of learning gains? Just as we answer question in science, we can apply education research to find out. Along with my colleagues, we developed the Microbiology Concept Inventory that was designed and validated to assess learning the full microbiology curriculum in the ASM curriculum guidelines. You can see the paper that my colleagues and I published here called the Development, Validation, and Application of the Microbiology Concept Inventory, and you can find the link to this open access paper in JIMBY in the chat. Our assessment tool was tested in dozens of diverse undergraduate classrooms around the United States, and you can use our tool in your microbiology course to identify concrete learning gains. Next slide. We applied our tool, our Microbiology Concept Inventory, to identify the most common student misconceptions in microbiology, and we published this data in JIMBY. You can see 3 common misconceptions related to informational flow in genetics in this slide. And you can see the name of the paper that we published in GIMB 2, and you can find the link in the chat. Our microbe author team responded to this data by rewriting passages from previous editions of the textbook to address these misconceptions that are backed by data. One example of a modification, data showed lots of misconceptions around the ancient endosymbiosis that gave rise to organelles and primitive eukaryotes. So we rewrote this passage to target these student misconceptions. Next slide. Another tidbit that you get with the instructor's manual is an extensive peer-reviewed test bank, and I'm going to stress the word peer-reviewed here. This is a collection of 264 multiple choice questions contributed by 52 authors from around the world. These questions are both for lower order thinking and higher order thinking questions. So here, this refers to Bloom's taxonomy, where thinking questions like identify or describe might be lower order thinking, or questions that ask the students to propose a science experiment or interpret data might be higher order thinking questions. All of these questions in our peer-reviewed test bank include example learning outcomes and answer explanations. And you can see one example question here on your slide where we have a question. You have the 4 answer choices that you could give your students. You have the correct answer choice underlined, and the writer included a note about why that's the correct answer choice. And I'll just stress again that these were peer-reviewed by the ASM education community, and you do get this as part of your instructor's manual. And I think that this will really help you find questions for maybe in-class clicker questions, or you could even use these on quizzes, so therefore saving you time in your class prep. Next slide. Additional resources that we prepared for you for Microbe 3rd edition is an extensive collection of recommendations that you could use for flipped classrooms. So flipped classrooms refers to the way of teaching where the students are given the content for the first time outside of class. So they might be reading the textbook, watching videos, and encountering the content for the first time outside of class. And then they go to class, and in class, they might have a review of what they learned outside of class. But then the key focus here in flipped classrooms is to do active learning during your face-to-face or during your synchronous learning times. So in Microbe 3E, we've collected a wide variety of resources for you for both the in-class active learning activities that you could use at any classroom modality, whether you're teaching online, hybrid, high flex, or in person. And those are called the Dig Deeper activities. And we also include a wide selection of recommendations for out-of-class self-guided learning where the student encounters the content for the first time. So in addition to reading the textbook, the student would also have recommendations for videos, podcasts, especially from the TWIV, the TWIMS, the TWIPS, from the American Society for Microbiology, and also text images and websites for student deeper learning. So I'll emphasize that these are all resources that come to you as part of the instructor's manual. So at this time, I'm going to turn it over to Elizabeth, and she's going to talk to us about scientific content.
Thank you, Rachel. This is Elizabeth Joyce, and before I talk about some of the new scientific content, I wanted to take just a moment to share how delighted I was to be invited as a part of this great author team for the new edition of Microbe. And the reason for this is that I taught my first undergraduate level general microbiology course in 2006. And as I was hunting around for a textbook to adopt, I was thrilled and relieved to find that the first edition of Microbe had just recently been published. It was written with a clear and engaging narrative that was accessible to students, and I think it did a great job of digesting a lot of information into concise concepts and basic principles. And I was especially appreciative of the inquiry-based approach, where the original authors didn't just focus on what we know, but how we know it by including guiding questions throughout the book. So we tried to approach this writing this new edition with the same spirit of inquiry and clarity, and incorporated some new themes and topics and ways of thinking about how microbiology impacts science and society. So on the next slide, at one of our earliest author meetings, we three focused on the concept of One Health. And this is an approach that really recognizes that the health of humans and animals and ecosystems are really closely connected and interdependent, and that our activities as humans impact the environment and climate, and these have a ripple effect on the health and well-being of entire ecosystems. And at the core of these ripple effects are microbes. You don't need to look any further than the dozens of disease outbreaks over the past few decades, and you'll see that the majority are zoonotic infections. Like this slide is showing Zika, and the outbreaks that occurred over a couple of years, the heat map showing where these occurred, as well as some of the symptoms, what the virus looks like, and some of the impacts it has had on society. We don't have to talk too much about other diseases like SARS-1 or SARS-2, for example, but their emergence is clearly linked to human activity. And of course, it's not just emerging diseases. The spread of antimicrobial resistance is a huge issue impacting many sectors of our society. So since human populations are clearly continuing to expand, the interactions between humans and animals and the environment are only going to increase. So we want to encourage our readers to view biologically important issues through a One Health lens as a way to understand this crosstalk between humans, animals, and the environment. And we've referred back to it frequently in the book. Our students will be the ones trying to address or solve these really complex problems. And this is going to require a collaborative and multidisciplinary approach, which is something that the One Health framework really embraces. So in the next slide, we also tried to incorporate some new material, and one of our new chapters in the book focuses on the gut microbiome. I think everybody in the entire world now sort of hears about the microbiome in popular media and culture, and there's a lot of understandable excitement about this dynamic and rapidly evolving discipline. It has incredible potential to impact human health. And while it's still in its infancy, advances in technology and computational power have really yielded robust evidence that's giving us a lot of insight into the nature of the microbial communities that live on us. There's so much coming out now on practically a daily basis, and our goal is not to really document every hot off the press discovery, because that would be impossible. Rather, we wanted to highlight some of the core concepts that have come out of this research, and also really wanted to illustrate to students the creative and interdisciplinary thinking that goes into doing this kind of work as a way to inspire and motivate them to think about this type of sort of meta research process. So we do cover factors that influence the development of the gut microbiome as sort of a paradigm for this, but of course there are microbiomes in all other parts of our body and other ecosystems. We talked a little bit about the data that supports the different functions of the gut microbiome, and really make correlations or point out the correlations between the microbiome and human health and disease. And again, really trying to highlight to students the creative and interdisciplinary approach to research in this field. Next slide. I'm going to hand it over to Michele.
Yes, Michele Swanson. Thank you, Elizabeth. It's been such a pleasure working with you. Your extensive experience in the classroom and also as a textbook author has really elevated our work, and I've learned so much. And Rachel, we were fortunate to recruit you to work with us in the second edition, developing the curriculum guideline framework for the textbook. And we learned from that experience how valuable it is to have a scholar like yourself really bring state-of-the-art best practices in education to the design of our textbook. So thank you very much. So a few features I'll highlight are the microbes at work features, where we, in chapters that teach kind of the basic mechanisms, we include these boxes to impress upon students that there are real world applications of this knowledge. And one example is, excuse me, using phage therapy as an alternative to antibiotics. And we include the reference to an open access article so that the instructor or the students can learn more. Other examples of the microbes at work include work from a recent MacArthur Fellow, Dr. Torres, who is featured in a Microbiologist at Work, sorry, last slide, please, who leveraged his expertise in toxin biology and bioengineering to develop new therapeutics that can stop MRSA in a mouse model. So we're excited to see where that goes. And then another example is Dr. Elizabeth Padilla Crespo, who is on faculty at the American University of Puerto Rico. And she read, when she was training as a PhD student at Georgia Tech, a report by the National Science Foundation about women, minorities, and people with disabilities in science and engineering, and really saw the data on how underrepresentation is holding back our field. So after finishing her training, she moved back to her native Puerto Rico, where she is applying her knowledge of detoxification of environmental groundwater contaminants to the studies of bioremediation and detoxification. So that's another example of applying microbes to real world problems. Next slide, please. We also feature how microbes can be part of the solutions to challenges that we face today in our Microbes at Work boxes. So one example is a really exciting advance in that fungi make these extensive root networks, and it turns out those mycelia are excellent substrates for lots of manufacturing. For example, they are heat resistant and also resistant to other stresses, and can be used for making insulation or for fabrics, leather that is made from plant material, from fungal material, rather than animals. So a couple of examples such as this, we think really it will excite our students and make them understand why it's so important to learn the fundamentals of microbes. Another example is we include a feature on using microbes to generate energy through microbes such as Shigella that through respiration will deposit their electrons onto a manufactured substrate that can then generate current. So this is another example of applying basic concepts like respiration, but in a real world example that we think will get students engaged and hopefully thinking about how they could themselves develop careers where they're addressing problems of concern. And on the next slide, we have also been intentional about including images that really capture the text that's discussing particular concepts. So for example, on the left side here, we're looking at images that were captured with the robotic arm Alvin, which is used in field work that Dr. Rachel Horak, our co-author, has participated in. And then the image on the right is SARS-CoV-2, which probably needs no introduction. These images are not only illustrating the simplicity and the power of these small viruses in our reworked virus chapter, but it also linked naturally with a box about the current pandemic. And we got to feature one of today's heroes, who was named a Times hero, the Times Magazine hero. And this is Kizzy. I am looking at my notes here.
Corbett.
Kizzy Corbett, yes, who trained at the NIH's Center for Vaccine Research and is now on faculty at Harvard's School of Public Health. But her work was instrumental in designing RNA-based vaccines for to protect against COVID. So the vaccines that we refer to now as the Pfizer or Moderna vaccine. So again, this illustrates how we've been able to incorporate diversity, equity, and inclusion, and also the application of fundamental microbial knowledge to challenges that we face in the world today. So I'll turn it back over now to Rachel.
Hi, this is Rachel Horak again. So we're going to stay on this slide for a minute. The fourth and final major new and unique type of content and feature in Microbe 3rd edition relates to elements that are intentionally designed to assist student learning. All the initiatives presented here are based in peer-reviewed evidence to increase student learning. So we use the principles from learning how to learn to guide what we put into Microbe 3rd edition. Next slide. Scattered in all parts of the book are features to help students think like scientists. These case studies pose current, real-world microbiology applications. Personally, I love using the subject of astrobiology, which is the study of the origin, life, and distribution and future of life on Earth. I love using astrobiology to teach microbiology concepts. So here is an example of an upcoming 2027 NASA mission to Saturn's moon Titan. This was presented as a case study for a chapter about microbial metabolism, and students are asked to consider what evidence for life could be used to identify if life occurs outside of Earth. Next slide. Part of the ASM curriculum guidelines asks students to be able to use data and evidence to draw conclusions. Students can develop these skills by examining original, published, peer-reviewed sources. In Microbe 3rd edition, we drew upon peer-reviewed sources, particularly leveraging the internationally recognized ASM journals, as you can see some examples here. And we used the peer-reviewed ASM image library, which is freely available, and a link's going to go into the chat box for that. You can see here on the left is a peer-reviewed image from the ASM image library that demonstrates a tube motility test. The image on the middle demonstrates an electron micrograph of an endosymbiosis. And the image on the right is peer-reviewed from PNAS about Vibrio cholera and phage dynamics. So we felt that it was really important that students see original data, original sources, and we leveraged ASM journals to do so. Next slide. You've already seen examples of microbiologists at work feature. These features are also serving the purpose to describe diverse career options for upcoming microbiologists, and they highlight current academic or industry research. We show one example here of Amy Richard Garlett, who earned a PhD in microbiology, did a postdoc, learned how to homebrew beer, and then changed careers to be a technician at a brewery, and she does quality assurance at the Dogfish Head Craft Brewery, which I'm pretty sure is the profession that I would pick if I did not love education so much. So I feel that these kinds of features are really essential to help students learn the broad range of careers available to them as scientists. Next slide. So we made sure to incorporate learning outcomes for all of the sections within Microbe 3E. These learning outcomes are presented in front of all the sections. So here's some example of learning outcomes in front of the section, how do bacteria and archaea modify their cell envelope? And you can see, looking at the example learning outcomes, that they span a wide variety of thinking skills. I'll read a couple to you. Provide evidence for how 2 cases of convergent evolution with regard to bacteria and archaea cell exteriors. So that could be a higher order thinking. One could be another learning outcome, and for a reason to explain why animal cells have receptors, that could be a higher order thinking. or you could have low order thinking skills, such as discuss how a slime layer allows cells to form biofilms. These learning outcomes spanned all the content presented in Microbe 3E. As a bonus, you could use these learning outcomes to design your own quiz questions, therefore saving you time. In the instructor's manual, all of these learning outcomes are aligned to the ASM curriculum guidelines so that you can categorize them and help the students better understand the concepts instead of just memorizing. We also ensure that we have check your understanding questions at the end of every chapter so that the students can do those outside of class to check their learning. Next slide. So with that, we're going to open it up for questions. We would love to hear what questions you have about our brand new Microbe 3rd edition textbook published by Wiley. Please put your question into the question box in the control panel.
Thank you so much, Michelle, Rachel, and Elizabeth. So yeah, as Rachel mentioned, we're moving on to the question and answer portion of the webinar right now. We've had some excellent questions coming through, so go ahead and keep on typing those into the chat box. We're going to try to get to as many as possible. So the first question that I'm seeing here is this book suited for general microbiology classes only, or is it applicable to upper level courses as well?
I can respond to that. It is designed in sections, and the first section covers the fundamental concepts. And then as you get deeper into the book, there is focus on ecology, environmental microbiology, and finally a section on microbial pathogenesis. So if students have already come in and they've got that solid foundation, then instructors can focus on some of the more specialized aspects of microbiology. And certainly the dig deeper activities, many of them are higher order thinking, so that those are also especially suitable for more senior microbiology students. So there's a lot of versatility in the book and a lot of instructor control.
Great. Next one we have here. So you talked about the ASM curriculum guidelines. As a reminder, there's also a link to that in the chat right now. How frequently are those updated, and sort of what does that updating entail?
Yeah, I'll take this one because I'm leading this effort. So the ASM curriculum guidelines for undergraduate microbiology were published in JIMBY in 2012. It was an international effort of educators that organized through the ASM Conference for Undergraduate Educators. It was a several-year effort that they took input from several hundred educators around the world. And we're going to work together with a task force. We're gathering this summer to start collecting our data and identify, are these still the concepts of all microbiology, or has microbiology as a field expanded to include concepts that we don't currently have? And we're also going to think about revising them of what are the other skills that our students need to be successful microbiologists. So we're currently undergoing the revision, and we will have that ready by the time next edition of Microbe comes out.
Awesome. So in addition to some of the things that you have mentioned and some of the elements added into the book, what are some other ways of incorporating these ideas around diversity, equity, and inclusion in the microbiology classroom?
I can start with this. I think, our classrooms are becoming more and more diverse, and I think simply opening up this kind of information and asking students to engage and just seeing how they are interfacing with this material is really helpful and insightful as an instructor I see this, even these sort of little tiny sort of micro experiences with students that have come from underrepresented backgrounds. I find that really insightful, and have struck up sort of casual conversations with them. So not necessarily even a formal educational strategy, but just simply engaging them and letting them know that their perspectives and insights are important and valuable.
And I agree, Elizabeth, and I'm having a flashback now. I was a biology major in college, and it wasn't until my senior year that my instructor was actually telling the stories of the microbiologists who would meet at particular meetings or at research labs, and talking about the people who did the work. And it made such an impression on me. I got much more interested, because it's one thing to read the black and white in a textbook, but to actually envision science coming to life. And so what I'm super proud of in this book is we have told the stories. So it's not just what the knowledge is, how respiration works, but actually who is now doing research and applying that to real world problems, and how they got interested in science. So the diversity of people that we represent, not only from the background they come from, but also what drove them to science, I think will really, I think that our students will recognize a piece of themselves in some of these stories, and be that much more interested in learning and hopefully pursuing a career in microbiology.
And I'll add to the conversation here that one of the parts of DEI is to recognize that our students bring with them a wealth of skills that may not be science skills, but they are skills that are embedded in real life, and those skills help them succeed in science. And we need to value those skills that the students bring with them. So I'll give you one example. I think that teamwork and the ability to have interpersonal skills and work with people that are different than you is very essential to being a scientist. So we wrote a feature for a Microbes in Society box about what it's like to work as a team in science, and how big and diverse these teams are, and the importance of these extra skills that may not be rooted in science, but these students that may have been traditionally underrepresented or marginalized and not being able to be scientists, those students are bringing with them these skills that are very valuable. And so we need to elevate and work with them in order for them to stay in science.
Yeah, and if I could amplify the team science, one, it's so true in my own experience as well. And I think many of the dig deeper activities that we've included are teamwork in the classroom or outside of the classroom, so that it can build those collaborative skills. And Rachel, I'm also reminded that your field work on really diverse teams out in the ocean, the North Pacific, studying carbon cycles and nitrogen cycles must have been exhilarating in that way. Could you tell us more about that?
Yeah. So I think I calculated I've spent over half a year at sea in several different types of voyages in the Atlantic and the Pacific, where I've done voyages where we did research with the deep sea submerged vehicle Alvin, and other manned submersibles. We've done lots and lots of open water, open ocean, nitrogen and carbon cycling. And so we bring that into the book, and I bring in pictures, images that I've taken at sea, and bring in some of those concepts so that we can have more representation of environmental microbiology and current issues in oceanography. So, of course, climate change is one of the quintessential scientific research fields right now. And oceanography, a lot of oceanographic questions target how do microbes influence and are influenced by climate change. And so I bring that perspective several times, into especially the biogeochemical cycling chapter.
Another great example of One Health that is woven throughout the book.
Mm-hmm.
Excellent. And actually, talking a little bit more about the dig deeper component. So there is a question, does this book come with separate online resources for the students? So I don't know if you want to talk a little about the dig deeper or the supplemental links and resources that are included.
Yeah, maybe Rachel, you could start with the resources that will be in the instructor's manual.
Sure. So the resources in the instructor's manual are all included through a link that Wiley will provide when you purchase the book. And they're provided just to the instructors, and the instructors are able to distribute those links as appropriate to their students. Does that help?
But they include links to the podcasts, and I'm pleased to be a member of the This Week in Microbiology podcast team, where we bring these fascinating stories and the stories of the scientists to a general audience. But that's one example. We also are careful to include a lot of open access review articles that provide more background in some of the topics that are covered maybe in a box. So instructors have the option of learning more themselves or pointing interested students or group projects toward these resources.
Yeah, awesome.
We also include a number of lectures that are available online, vetted lectures from thought leaders in our field, National Academy members, for example, if you would like to provide out of classroom background material for students then to launch an individual project or to better appreciate the topic that you're covering in class.
So I'm synthesizing a couple of questions together here. There's sort of one question asking, it seems, a little bit more about what's covered in the new microbiome chapter, and also related to that, are other newer technologies or areas of study like CRISPR, or how deep is the coverage on COVID-19? If you can speak to those.
I'll start with that one. So the microbiome chapter, I mean, I started writing that and I just kept going on and on and on. It was fascinating, and you could write an entire book on the microbiome. We pulled back a little bit and decided to focus on these sort of core concepts as a way to illustrate that you can do this large-scale biology, this kind of meta approach and computational approach, very interdisciplinary and collaborative approaches to study this incredibly complex system. So I think that was it was deliberate in the sense that we wanted to sort of showcase a couple of really key studies that have been published as a way to show how students can interpret some of the, we pretty much interpreted the story for them, but because they're complex studies, and we paired with the chapter will be these dig deeper questions where we're asking students to actually analyze a paper and really think hard about not only the scientific data and how they can convince themselves that this is something that is believable. I mean, all these things are quite complex, but also considerations of ethical issues about doing studies on microbiomes and the implications for communities, trying to infer conclusions for all communities. So I think that there's a lot to offer in the microbiome chapter other than just the factoids about the microbiome, which are fascinating. The second part of the question, so I actually have the privilege of working at UCSF, where there's amazing faculty, and one of the wonderful faculty members in my department is a CRISPR expert who hails from Jennifer Doudna's lab, and I consulted with him, so I feel like we have the most up-to-date information that we've included, and it really updated that material in the chapter. And we also have a microbiologist at work focus on Dr. Doudna. So we spent a lot of time trying to elevate and showcase the new technologies, because these are things that if students become interested in research, they'll certainly probably be using those. But I also wanted to make sure that they had the framework, an inquiry based framework way of thinking, because there's going to be new technologies coming down the pike that we don't even know about yet. And if they can sort of use these as a way to capture their imagination and really push themselves to think hard about how they can approach a scientific question, I think that we'll have succeeded.
As for SARS-CoV-2 and COVID, we were updating this book right up until our publisher's deadline with new data about what we were learning about SARS-CoV-2. So we included as much up to date as we could, and we take a One Health approach to discussing COVID-19. Anything else you wish to add, Michelle?
I can't resist. So this is the new Microbe 3E. I'm delighted to share that it is now a book. And speaking of what kind of coverage have we given to some new topics? These are the 2 Nobel laureates that worked on, that discovered CRISPR-Cas.
Awesome. Always nice to see the actual book there.
Yeah.
I want to address, there's a couple of questions in the chat that have been coming in asking about whether figures from the books are available or are there PowerPoint slides. So yes, for everybody asking that, as part of the instructor materials, PowerPoint slides are available for each chapter that feature all of the beautiful images throughout the book, all of the figures and the tables that you can use for your teaching. Yeah, so let's see, just looking through the questions here. Just a general one, kind of what sets this book apart from other microbiology textbooks that shall not be, or they shall be named?
I'll start. When we had our first author meetings, we looked at the data and saw what books are in use across the world. And a lot of them are really impressive, comprehensive reference manuals where you can learn about anything in a lot of detail. But when thinking about actually teaching microbiology to early students or to more senior students, we realized that that textbook is not useful. Rather, we wanted to tell the stories and get across the fundamental concepts and in a visual ways and really engage the students. So we don't attempt to be comprehensive. Rather, we cover the major components of microbiology, from building a cell, fueling a cell, dividing a cell interactions, etc. But then through the boxes and through some of the exercises, give the opportunity for students or instructors to engage more deeply and at an advanced level.
And
Anyone add to that?
Go ahead.
Yeah, I'll go ahead and add that we really use the data about what we know about how students learn and how students know microbiology and what those common misconceptions are to frame our writing. And from my knowledge, it's the only book that does that. So we took the data about how students learn and knowing those misconceptions to reframe the book and rewrite passages to directly address those misconceptions.
And I'll just add one other comment that I think as any students, no matter what field they're in, they're always going to need to be pushing themselves to learn. So we really wanted to foster this sort of inquiry-based approach and way of thinking about science. It's not a static endeavor. You know, a textbook, you can update it online, but a textbook is a static thing. And while that can serve a really important, as a really important resource, we really wanted to push students to know that they themselves can go and investigate, and they can go look up things on their own. And we provided a framework and enough of a foundation for them to be able to go and do that, we hope.
And as another example of that, we also know from the literature that active learning is a much more effective method than having the sage on the stage. And that's why we've included activities with each chapter for in-class activities where they can apply knowledge and really learn how to communicate as scientists, learn how to do research using vetted resources to learn about a topic that interests them, and also science communication. So activities where they are encouraged to put together a 3-minute presentation for their class on a topic that they thought was interesting related to that week's subject matter, or to write a newspaper style article, maybe for their college newspaper, that describes a really cool bit of science that they've just learned about or that's in the news.
Awesome stuff. Another just kind of general question that's come up that I'm just going to address now, which is the instructor manual like a separate book that folks have to buy? So no, the materials are all online and they're made available to the adopters of the book. Here is a pretty interesting one. So much of the world's population thinks about microbes as being bad or harmful. So are there aspects in the book that kind of help to change this type of thinking in young budding microbiology students?
I'll bet your microbiome chapter, Elizabeth, goes a long way.
I'm going to argue that most of the book actually talks about the beauties and wonders of the myriad microbes in the world. Of course, we do have a couple of chapters on organisms that can cause disease, but we really try to frame this book as a way to introduce microbiology in its entirety to students. So ecology, of the human microbiome, and really showcase how most microbes are, we may think of most microbes as being benign, but I think that's just because we don't know how wonderful they are. And I think that as we learn more and more about specific organisms or communities of organisms, we'll actually see that they're beneficial. They're here for a purpose. And so I think we try really hard to showcase that and highlight that.
Yeah, and they're small but mighty. Once we understand their biochemistry, we can then leverage that to address real world problems. So I'm reminded of our chapter on the cycle of elements. We have a microbiologist at work feature on Dr. Jennifer Glass's work. So she's an associate professor at Georgia Tech, and she is interested in the biogeochemical cycles and astrobiology, and is able to set up in her lab conditions that mimic the early stages of life on Earth, microbial life on Earth. And she's using this model to understand how microbial metabolism influences greenhouse gases, which of course affects climate change. So again, another example of not only what the science knowledge is, but why it's important, who is doing the work, and that it's one health, it's all related, microbes and the air we breathe and the temperature of the planet.
And even viruses, we even showed examples of viruses that aren't necessarily bad. So we gave one example about the potential for phage therapy in our presentation. And then we also talk about viruses in the ocean. And I think that those are not talked about enough, but they're essential to the oceanic carbon cycle. So we talk about the viral loop. So I think there's examples of microbes that are beneficial throughout the entire book.
Another just general, everybody who's asking questions about pricing or how to get the book, we've just placed a link into the chat there that is the link to the book's landing page on wiley.com. So you can see all of that information right there. Let's see, here's one about the activities. So have you used any of these activities yourself during teaching, and are there any that you think are most effective?
I was fortunate to teach a first-year seminar here at the University of Michigan. So these are for non-majors, it's their first term on a college campus. And one exercise that I had a lot of fun with was I'd give them a brief introduction to a particular topic, and then give them a couple podcasts that they could listen to, and then encourage them to write as an assignment just a one-page description, like for the college newspaper. And I think that my goal there was to just get them excited about that microbes are everywhere and they impact every aspect of our life, whether it's the food we eat or the water we drink, the air we breathe, and also developed their writing skills and gave them confidence in beginning to do more critical thinking as would be expected at college.
And I currently am teaching in a professional school, so medical, dental, and pharmacy students. So I adopted some of the applications that we showcase in the book, so team-based learning, to incorporate some of the pathogenesis that was appropriate for the groups that I was doing this with. So nothing exactly precisely like what's in the book, but an adaptation of things that we have developed for the book.
And I'll add to, if you're interested in learning more about activities for the undergraduate microbiology classroom, I'd encourage you to take a look at a link that was posted into the chat. It said for more ASM content and resources for undergraduate educators. Take a look at that. The ASM offers a huge variety of resources to help microbiology educators do their job more effectively. So come part of the community, come to our education conferences so that you can get more ideas from other educators to implement in your classroom.
Yeah. And I'll also add that at the upcoming ASM Microbe meeting in DC in June, the book will be in the ASM bookstore, and Rachel and I will be available also to meet with people and talk and hear your ideas and your needs.
Yeah, we're really looking forward to that, having that interaction there. There's been a couple of questions I think we just want to clarify about whether this is a lab manual or there's a lab component to the book. So just let you guys.
There is not.
Yeah, techniques are mentioned and covered through the book, of course, but it's not a manual and there's not an associated lab manual with it there.
That's right. So we really cover some of the most important and universally taught techniques, such as gram staining, right? But the purpose is not to teach the manual protocol of gram stain. It's to teach the concepts of the gram stain in the overarching view of how a cell exterior is.
And also sophisticated whole genome sequence analysis and how that has really revolutionized our thinking about microbial evolution, et cetera. So classic techniques and also state-of-the-art techniques.
All right, and if anybody is having trouble clicking those links in the chat, there will be a follow-up email, and we'll be sure to include those links in that as well. So we're getting close to time, I guess. Elizabeth, Michelle, Rachel, is there anything else that you want to say to just kind of round out this awesome webinar?
We look forward to hearing from our adopters and their experiences in the classroom, and we'll certainly, it'll be energizing for us, but we'll also apply that for future work.
That's absolutely true. We encouraged our Microbe 2E adopters to give us feedback, and we talked to them extensively of what would make a better textbook for their students. So we do want to engage with our adopters and our broader microbiology education community to design the best textbook that works for the most students.
And to make it affordable and accessible. And we appreciate the partnership with ASM Press and also Wiley to do that. Megan, could you remind our listeners what materials you'll be sending them after? They'll get the slide deck and...
Yeah, absolutely. So as a reminder, the recording of this presentation will be emailed to you within a few days. And you can right now download in the little handouts section of your control panel the PDF of the presentation that we saw today. All right, so we're coming up right on the hour now. So I just want to thank again Dr. Swanson, Dr. Joyce, and Dr. Horak for all of the excellent information that was presented, as well as everyone in the audience today for all your thoughtful questions and for taking the time to join us. And with that, we will say goodbye. Thank you.